Okay, so it's my pleasure to introduce our speaker for today. So Dr. Erick Boy has been Chief Nutritionist of the Harvest Plus Biofortification Program at IFPRI since 2008. And he holds a medical degree from the University of San Carlos in Guatemala and a doctoral degree in human nutrition from here at UC Davis. He has over 30 years of experience conducting and managing nutrition research at global agencies like the Institute of Nutrition of Central America in Panama, the Micronutrient Initiative, and Harvest Plus, aiming to tackle hidden hunger in low and middle income countries. His research has focused on micronutrient bioavailability, efficacy of biofortified staple crops and food fortification, zinc status biomarker, and associations between zinc deficiency and cardiometabolic disorders. He's also served as a technical committee member for a number of different organizations and numerous other service positions. So we are really glad to have you joining us, Erick, and I will let you take it away.
Okay, thank you. Thank you. And it's a real privilege to have been invited and to be able to share some of the work that, because I went to UC Davis, I was able to do during the past 20 years. So I will try to be brief and hopefully clear about the research trajectory and future perspectives in research for biofortification of staple crops. And this is, of course, the perspective of conventional plant breeding for biofortification. I will not touch the subject of genetic modification or genome editing for the same purpose. That means to biofortify staple crops. So the research that has been carried out with many top-of-the-line collaborators around the world, North and South continents, obeys a very well thought out plan that was in place before I arrived in Harvest Plus, and it goes from 2003 to this day, starting with the discovery phase where the nutrient breeding targets for the different crops were agreed upon by nutritionists and plant breeders. There was a lot of investment in vitro Keiko2 cell bioaccessibility, bioavailability studies, mostly done by Cornell University. And there was an upgrading of the technology that went into the screening the different crops materials for higher nutrient content and to be able to have very high throughput and cost-effective screening of thousands and thousands and thousands of accessions or materials available in the gene banks of the CG system. After that, the first efficacy trials and several bioavailability studies with humans were carried out, and the country and crop combinations were selected, looking at countries where these major staples were consumed as the main source of calories. And the original idea was to start doing the efficacy trials after the materials, the varieties were released into countries, but because of donor pressure, the cart was put in front of the wheel, and so the study, efficacy studies began before many of the released varieties had achieved the maximum level of biofortification. So today, most, if not all, of the investment in biofortification is going to scaling up in countries in Sub-Saharan Africa and South Asia, Latin America, as with many other donors and research efforts, has fallen off to the side pretty much. And to summarize the area of research that is not within my purview or area of expertise, the socioeconomic and crop development research that was done during this 25 years contributed significantly to raise funds by carrying out and publishing ex ante and post exposed benefit cost analysis, which also helps to guide the plant breeding efforts, say to select which priority micronutrients, so iron, zinc, and provitamin A, which crops and in which countries would we have the best opportunity to prove the concept that biofortification can indeed improve micronutrient status in a replicable manner. There were a lot of farmer and consumer surveys, as well as monitoring systems established to estimate the adoptions of these varieties once they were released by the ministries of agriculture in countries, and these models had not been published until very recently, when because of public and private pressure, our monitoring team finally submitted the methodology paper for publication, so that we can with more confidence talk about the numbers of people being reached today. So going back to what I know more about, it's the food and nutrition research behind the proof of concept that biofortification is a viable, cost-effective intervention to tackle micronutrient deficiency as a complementary, not as a silver bullet, but as a complementary intervention. So this research had basically 3 stages. The first one was the feasibility stage, where the question to be answered by the research was whether the crops could contain enough micronutrients through plant breeding, and whether these micronutrients would be available for our bodies to absorb and use and utilize. So this meant we had to manage studies on dietary intake in the target countries, as well as nutrient loss during processing and storage of these crops, and bioavailability or bioefficacy studies to make sure that these nutrients were not only not lost too much, but that the nutrients that were left were bioavailable. And this justified the next stage, which would be the proof of concept or efficacy study stage, which, as you know, are very expensive and complicated. And that stage only included 2 to 3 efficacy trials in different countries with the different crops. And the final stage, the effectiveness assessment, didn't really get finished because of lack of funding. So instead of doing an effectiveness trial or 2 per country crop combination, we were able to only do 1 trial per nutrient and crop combination. And this is the scorecard. Everything that is in green signifies a type of study and number of studies that were completed since between 2003 and 2023. And as you can see, most of the studies on dietary intake, nutrient retention, bioavailability, and efficacy were completed. And for effectiveness, like I just described, only 3 studies were done. And for zinc maize, we decided not to do any further analysis, because we found out early with a study by Dr. Hambidge in Zambia that most of the zinc in the maize would be lost during typical processing, milling of maize in sub-Saharan Africa. So the first issue to tackle or problem to study was whether breeders could get the micronutrient levels in the staple crops high enough so that when consumed, these crops would generate a measurable change in nutritional status. And the consensus between breeders and nutritionists then was that the bars in green, which represent the increase necessary to produce these effects, that these bars could be indeed achieved from going from 55 micrograms per gram of iron in beans, the first column, to 94, almost double the concentration of iron. Was it possible? And when all of the material was screened, all of the material, the seeds in the gene banks was screened, it was found that the biological variation or the natural variation of this nutrient in these crops allowed for breeding to achieve the levels that we see here. So for beans and pearl millet, the iron levels would be significantly higher, and importantly in nutritional sense. However, for zinc crops, the zinc increase could only reach an additional 12 micrograms per gram. So that was the top that the breeders said they could achieve. And even though that's not what nutritionists said would be enough to produce a significant improvement in zinc status, well, we accepted that and tested material with that new level of zinc. For provitamin A crops, cassava, maize, and sweet potato, it wasn't a problem to get the alpha beta carotene and beta cryptosantine levels up to a concentration that would produce a measurable change in vitamin A status. So because that natural variation allowed for the breeders to produce materials, seeds and planting materials with the higher levels of these three nutrients, it has been estimated that as of last year, over 400 different varieties had been released into countries by agricultural sector in over 30 countries, and there were many other varieties in the pipeline, being tested for further future release. And the only problem was that we didn't anticipate that the funding to continue doing this called targeted breeding was going to end. And that had a big impact on the size and dynamism of the pipelines at the CG centers in charge of each of these crops, as well as the centers for agricultural research in the countries. So as of last year, this was the number of varieties released and the countries that were commercializing, farming, consuming them. And so it was possible to reach these levels, but then we also had to study how much of these micronutrients would be lost during typical processing in rural villages by smallholders in sub-Saharan Africa and Asia. So for rice, studies found that if you parboiled and then milled the rice extensively, like they do in Bangladesh, with a loss of 16-18% of the material or the matter of the grain, you would lose, and that's the colored column, and the bottom is the worst case scenario. So if you parboil with extremely hot water and a lot of water, excess water, you would only retain 7% of the zinc. But if you process the rice so that you didn't parboil it and you milled it at 10% degree of milling or 7%, then you would be left with 85% of the zinc in the grain, and that would be sufficient to conduct the bioavailability and efficacy test, because people consuming a rice that was not parboiled and was milled moderately and not severely, then rice could provide enough zinc to improve zinc status. This is zinc. Okay, and this is the story with maize. And as I preempted earlier, and the maize can be processed in basically two ways, like they do in Central America, where they nixtamalize the maize and they lose very little of the coating of the seed and the germ. Most of the zinc remains in the food, the tortilla. So 96% of the zinc remains, and you can provide 64% of the EAR for a child between 2 and 5 years of age. But if you process the maize as they do in South America for arepas, where you basically get rid of all of the germ and all of the pericarp, then you're only left with 35% of the zinc, and there is very little contribution of that maize to zinc nutrition. So it matters where you and how you process the maize. And so for Central America, biofortifying with zinc makes sense, and therefore that's why there is a very successful NGO in Guatemala and El Salvador commercializing a high zinc, high QPM type of maize in those countries, because a lot of this mineral is retained. And then for provitamin A crops, in this case maize, which is the worst case scenario because of the length of time that maize can be stored, the loss of carotenoids can be quite significant. So if you store the unshelled grain for 240 days, so almost a year, you end up with maize that contains only about 35%, 40% of the original provitamin A carotenoids. And so you can see the average for each type of processing. And no matter what type of bags you use, unless you use vacuum sealed type of packaging, the provitamin A, the carotenoids will degrade as that is the natural process of provitamin A and vitamin A compound. So before I go into the bioavailability results, I want to say that all these nutrient losses and the bioavailability results were all adjusted for when that level of micronutrient being targeted was determined and sought after. So the levels that I showed first, like 15 ppm for provitamin A in maize, or 100 ppm iron in beans, these levels already adjust for these losses. And so when the cooked food is consumed, enough of the nutrients is absorbed that we expect to have a nutritional effect, measurable nutritional effect. And the bioavailability results for zinc and iron different crops, always comparing to a control that's not biofortified, that means it had lower micronutrient content, it showed that there are really no significant difference in the percent absorption. But because the amount, the absolute amount of micrograms, additional micrograms of these minerals in these crops is higher, then at a given percent absorption, the total amount absorbed would be significantly higher. And this the same message for provitamin A carotenoids, the bioconversion of provitamin A carotenoids into retinol in the 3 crops that contain provitamin A proved to be lower than expected. So instead of 12:1 beta-carotene to retinol conversion equivalency, findings for cassava, maize, and golden rice show almost twice as efficient an bioequivalence of beta-carotene to retinol. The only crop that still required about 12 or 14 micrograms of beta-carotene per microgram of retinol is orange sweet potato, and this is a study that Marjorie Haskell did in Bangladesh. And so this is what I was talking about. So the adjustment for these losses during cooking or milling and storage, and this is the vitamin A maize example, were taken into consideration when estimating what percent of the estimated average requirement for a child or an adult woman, non-pregnant, non-lactating, would be provided by consumption of these crops as staples. So that means 50 to 70% of the daily requirement of energy. And so these are the factors used for that adjustment, and those are the targeted percent EAR contribution from this crop, and we have that for all the crops. So after all this, let's say, basic research, then we were justifiably in a good position to invest in the efficacy randomized controlled trials and effectiveness randomized controlled trials to test whether regular consumption of these biofortified foods when compared to consumption of controlled crops would increase micronutrient intake absorption and therefore a nutritional impact. And so here I'm summarizing, and let me see, let me check my clock to see how I'm doing with time. Okay. So provitamin A crops. These 3 crops, maize, cassava, and sweet potato, when biofortified, can provide between 50 or 100% of the requirements of this vitamin for children. The 100% is only applicable to sweet potato, and the 50% or from 40 to 50% is applicable for cassava and maize. As mentioned before, the conversion of provitamin A carotenoids to retinol is highly efficient in the three different crops. And all the studies that have measured serum retinol or total body retinol have shown that consumption of these crops does have a measurable impact on these indicators or biomarkers. And one study where breast milk retinol was studied in Zambia proved that the levels of low retinol in breast milk decreased in the mothers that consumed the high provitamin A maize. So there were multiple biomarkers and functional indicators measured in these studies. And to summarize, maize, when consumed regularly from 3 to 6 months in Zambia, improved night vision or dark adaptation, as measured by dark adaptometry using goggles and hooked up to a computer developed at Johns Hopkins, and it improved milk retinol in the mothers. The sweet potato in the two largest effectiveness studies, 20,000 households each, did not improve serum retinol. I mean, that's not correct as stated here. But it did improve serum retinol only in the children under 5 that had deficient levels when they began the trial, but it didn't improve the serum retinol in mothers. And in a secondary analysis, which Alan de Brauw from IFPRI did with the Mozambique results, he found that the children who had consumed the sweet potato for 2 years had a lower number of diarrhea episodes, and the episodes lasted less time in children under 5, and even more so in children under 3 years of age. And so that's an unexpected finding, and it is an inconsistent finding even in vitamin A supplementation trials, the impact on diarrhea, which I think has to do with the contextual matter in each of the countries. And in Kenya and Nigeria, where cassava was tested with children between 4 and 8 years of age, in both studies, mean serum retinol was improved when compared to controls in different analyses. The iron crops were tested, iron beans in Rwanda with university students, female university students, in Mexico with boarding school rural students, and for pearl millet in Indian boarding school adolescents as well. So in both the Rwanda and India studies, the results were impressively consistent. Iron status improved, more so in India adolescent than in adult women. Serum ferritin improved significantly, and total body iron improved in both studies. Cognitive performance, and particularly the efficiency of memory and attention functions, improved in the study subjects in both studies. There was also significant relationship proven between iron status improvement and physical work efficiency in the case of Rwanda, or the level of physical activity in the case of India. So all the important functional indicators of iron status and the biomarkers, improved in the right direction in a very consistent manner. In Mexico, there were no measurable effects, mostly because the children only ate almost half of the beans that they were supposed to. They had a very good menu, and they were not really iron deficient, not importantly so. That, and then an unequal distribution of respiratory infections during the trial really confounded the study, and we didn't see an effect in the Mexican school children. When we come to zinc biofortification, that's where we start hitting bumps with the sensitivity of the biomarker, namely plasma zinc concentrations, which we consistently found unresponsive in five different trials. So 2 efficacy trials in India, actually 3, India and Pakistan, and 1 effectiveness trial conducted independently by Nikki Lowe from the UK in Pakistan. In none of these studies did we see a response in plasma zinc. In one of the studies, serum ferritin went up, but not plasma zinc. And that's because when you increase zinc concentration in some of these cereals, iron also goes up in the same direction. It goes up, but not in the same proportion. The only study where we saw an improvement in functional indicators of zinc status was in a 3,000 household study in New Delhi, where the time spent with common morbidity by children and women in the intervention group was significantly less than in the control group. But even in this study, plasma zinc concentration didn't change. In the other studies where samples were analyzed for colonic microbiome, for DNA ruptures, and for fatty acid desaturase activity, there were no significant differences in them. The one study with rice zinc in Bangladesh, which lasted for 9 months, we thought that by extending the feeding time, we were going to see improvement. There was no improvement in plasma zinc, and there was a very modest improvement in linear growth, but it was a time by treatment interaction that was significant, and no change in microbiome metabolites or fatty acid desaturases, as I just mentioned. So with zinc, problems with plasma zinc concentration. And these are the results for the morbidity, the time spent with morbidity episodes by women and children in the Delhi trial. So significant % of time with lower risk of pneumonia, vomiting for the children, and fever for the mothers. So my take on this efficacy results with zinc crops is that I rather look at the absolute amount of zinc being absorbed when you compare intervention versus control, rice, wheat, or maize. And all of the studies conducted with stable isotopes with rice, wheat, and maize have shown that a significantly larger amount of zinc is absorbed when the biofortified crop is consumed. And that gives me pause to expect long-term positive effects on zinc nutrition, immunocompetence, and the other functions related to zinc status. And this is a study that another one of our prestigious UC Davis alumni recently submitted for publication, and it just goes to show how much of the zinc in the biofortified, in this case rice meals, was absorbed compared to conventional rice. And you can see at the chart on the extreme right side where that blue arrow is. So conventional rice provided 1.1 milligrams of zinc absorbed. Conventional rice plus zinc added to the meal when it was being consumed provided 1 milligram total absorbed zinc, and biofortified zinc provided 1.4. So 300 micrograms more zinc daily for children of below 3 years of age. That would be significant over time, especially during this 1,000 window of opportunity. And so we're looking forward to seeing this paper published. And this was Munirul Islam from Bangladesh ICCDRB. So after having completed all of these studies and taking stock of the results, I think that biofortification, the concept of biofortification being an alternative intervention, food-based intervention to tackle micronutrient deficiency and to tackle micronutrient intake inadequacy is proven. And it is really an intervention that targets the poorest in rural areas where these crops are grown and consumed, and where other interventions cannot reach or are not accessible consistently. So I think this is a foundational type of intervention at the very basis of the food pyramid, and it is worth pursuing with more research and development of varieties and crops that have even higher levels of these micronutrients, which is possible. And so, during all of this research, new hypotheses and ideas came up, and some of these were pursued, and some of them are not exclusively linked to biofortification, but they are linked to the more global nutrition, micronutrient nutrition effort. And one of those is the meta-analysis conducted by an ex-Cornell graduate and myself on the impact that low-dose, long-duration zinc supplementation, which would be comparable to zinc from food that has high zinc contents, can improve risk factors for diabetes and cardiovascular disease risk factors. And so this meta-analysis led us to, okay, well, let's now test this in vivo, and let's continue to estimate what the impact of zinc inadequacy could be on the days disability adjusted life years generated by deficiency in the developing world. And so the disease burden estimated from zinc deficiency through high fasting blood glucose, that means high risk of type 2 diabetes, was calculated for all countries and regions. And it was estimated that the burden of disease due to zinc deficiency through type 2 diabetes was much higher than that associated with diarrhea in infancy and early childhood. And that also gave us more justification to pursue this further. And well, this is a summary of those estimations that were published like 2.5, 3 years ago. And so you see the meta-analysis shows that zinc deficiency is associated with a much higher, also having high fasting plasma glucose. At least this is the case for Bangladesh. And it's the same thing for other South Asian countries or sub-Saharan African countries, where the epidemic of diabetes and cardiovascular disease is just becoming untenable. So with all of this information, important zinc researchers like Andrew Hall, who's a friend and colleague at UC Davis, and other very important researchers like Zulfikar Bhutta, Nikki Lowe, Martin Broadley, they all came together and presented all this information in a call to action to tackle zinc deficiency, more so than countries and agencies are tackling it now. There are very few country programs where zinc fortification or zinc supplementation beyond zinc for diarrhea is being implemented. And why? Well, because zinc deficiency control interventions don't produce immediate effects, which politicians always want, right? Or effects that you can say without an iota of doubt that is caused by the zinc intervention and investment. So there was a call to action for more investment in zinc interventions, and we funded a study in Pakistan in which zinc biofortified wheat flour, post-harvest fortified wheat flour with zinc, and non-fortified flour were fed to adolescent boys and girls and young women, 1,000 of them, 4 groups of 250, for almost 5 months. And we were interested in the effect that this additional zinc or not would have on metabolic health. And The preliminary results do show that a significant improvement in fasting blood glucose in both the groups that consume either fortified or biofortified zinc wheat bread. And so, this is, I think, the first proof of concept that food-based zinc interventions can improve what supplements also have been shown to improve, and that is risk factors for type 2 diabetes. Another, how am I doing with time, please? Okay, so I'm going to start wrapping up. Another, I think, important contribution that the research conducted by our collaborators has produced is this study by Andrew Hall and Victor Ravoy, et al. It was a small study, but what we're trying to do is to test these wheat varieties that were supposed to have higher phytase activity levels naturally, and to test whether these phytase in the wheat could decrease the phytate content and improve dietary zinc absorption. And we proved that the enzymatic activity wasn't enough to continue to breed for these higher phytase levels. And that's important. That's important information, because why continue to invest in breeding for something that's not going to be efficacious? And this study also proved that fermentation is a much better way to improve zinc absorption, and that has been known for a while. But that's a study that hasn't been published, and I think an important contribution for plant breeders interested in higher zinc absorption in wheat. Another study that's tackling the same constraint of bioavailability of minerals from food crops, as these are consumed in developing countries, is this study that Andrew Hall is also carrying out at UC Davis with courageous volunteer students, and in which we're testing the impact of lowering the phytate in beans, or lowering or changing the polyphenolic profile of the beans. So we're testing a bean that has been developed to contain a low phytic acid mutation, from Italy to Colombia and then back to Davis, and also testing it against white or other colored beans. And we hope to have results and a publication from this study at the end of the year. And these results will help also advise plant breeders where to concentrate their efforts to develop bean varieties that have significantly higher bioavailabilities for zinc and iron. We're measuring the absorption of both the nutrients. And then beyond single crop and nutrient approaches, in southern India, we tested multiple crops as part of a menu to improve the dietary quality or micronutrient density of the food that these children were consuming as part of the national food supplementation program for children under 5. And as you can see here, the green bars illustrate how much higher zinc, iron, and vitamin A intake these kids experienced during the 6 months of the trial. Unfortunately, we don't have results on biomarkers for this study. I'm pushing my friend at Cornell University to publish, but you know how these things go. Other studies come along, and we'll continue to try to get this one published. And last but not least, a study we did in Guatemala where sugar had been fortified, not biofortified, with vitamin A for over 30 years. We examined the livers of almost 100 cadavers that died from non-disease causes, like only from trauma. People who had lived in Guatemala all their lives, aged 16 to 60, and we measured the liver retinol, and we also looked at the liver, whether they showed signs of toxicity or not, and the results are here. And you see that in the graph, in the scattergram above, you can see that most of the values for liver retinol are beyond or above the 1 microgram per gram, micromole per gram liver retinol value. And that was considered the hypervitaminosis cutoff level for vitamin A. However, there were no changes in the histology of the liver. So biochemically, these subjects have too much vitamin A, but histologically there were no signs of toxicity. So this information will be used by the Guatemalan government and sugar producers to discuss whether to lower the levels of vitamin A in sugar, or whether they also need to monitor the levels of vitamin A in other foods that people are consuming that are also fortified but not regulated in the country. So we have reached, according to last year's 2023-2024 estimate, over 300 million people with biofortified staples. Some fully biofortified, others not. But this is the number of people that are growing, eating, or just buying biofortified crops from the market. And so what we don't know is whether this has any impact on population nutritional status, and that's a big challenge that we will be facing in the future. And we don't know how to unravel this, because most of these crops are not traceable visibly, and so we need to also develop ways to identify these crops through simplified technologies. I will leave the climate impact on micronutrient contents to the side, and just finish by flashing this list of improvement needed in biofortification to improve the impact of that these foods can have on nutritional status. So improvement in carotenoid stability, specifically for maize, and determine whether provitamin A carotenoids decrease aflatoxin exposure in maize, improve bioavailability of iron and zinc in beans, revise the baseline micronutrient levels for beans in Africa, which publication from Cornell denounced as not being correct, and so we're looking into that. And also continuing to invest in zinc biomarkers, biomarkers that hopefully can respond more sensibly to food-based interventions. So I will end with a summary of the research results on the 3 groups of biofortified crops. I think they are all positive and encouraging. And the challenges now lie on how do we scale up, and how do we scale up credibly, and how do we keep breeding for more high quality crops that farmers want and consumers will buy and eat. And with that, I want to say that all of this was only possible because of researchers like yourselves, institutions like ministries of agriculture in all of these developing countries, and of course the donors that have made all of this possible through the years around the globe. Thank you very much.
Thank you so much. That's just amazing to see the history of decades and decades of research in the area, and to see how it's continuing too. So thanks so much for sharing that with us. And we do have time for some questions. So I see Steve is ready with one. Okay, I will let Steve go.
Erick, you didn't, as an economist, I feel compelled to ask the cost question. I think in a non-resource constrained world, I think everybody would vote yes on biofortification as perhaps the platform on which we build other interventions. But we don't live in a resource unconstrained world. So we have to make some hard choices about the resources that we are going to be able to secure and use to deal with micronutrient deficiencies and inadequacies. So could you tell us a little bit about the cost side of biofortification? You don't have to say much about the history and all the millions that went into developing that. We know the upfront costs are huge. But once they're developed, are these biofortified crops more expensive to produce and therefore fetching higher prices in the markets? Or what else can you tell us about kind of the economics of these crops?
Yes. So there is an upfront investment in the research at the breeding centers. But that happened over the first 20 years, right? So now the centers are supposed to have their own budgets, and and to have integrated these nutrient traits into the breeding that they normally do for higher productivity, pest tolerance, etc. And the testing at the national research centers is done as part of what they normally do with any variety that's being studied for release in any foreseeable future context, like what virus is going to be prevalent, et cetera, et cetera. So these are costs that the centers normally absorbs. And so that, all the pipelines are already there. They should just continue to breed for higher nutrient for markets where it makes sense at no additional cost. And then when they get to the market, what we've seen is that at the beginning, when the production is lower than the demand, of course, retail sellers would hike up the price, and that creates problems because at the beginning, people don't have access economically. But as we saw recently in Kenya with the beans that are being commercialized there, once the prices were equaled through market forces, then access was no problem, and there was really no difference in cost or in price for the biofortified versus the non-biofortified beans. So in short, upfront investment done, continued investment should be part of what the institutions do, and the price to consumers cannot be increased. There is no justification to increase them because these varieties should be as productive as the ones that they are replacing, and without additional inputs. And not necessary. It's necessary to add new agronomic or economic inputs to produce these crops. Yeah.
Thank you. I think I saw another hand up. Yeah.
Okay. Thank you for your presentation and the information you shared related to biofortification and nutrition outcomes. My question is related to the prioritization of focus for biofortification. As I heard from the presentation, there are around 400 groups selected for biofortification. So if these groups are really representative to all countries or based on the country-specific diversities as different countries are using different biodiversity focus. And the other question I have is related to the biodiversification and climate change. As you know, several countries are facing dual challenge of malnutrition and climate stress. So how these strategies work in this?
Those are several questions into one, but I'll try to address them. So yeah, these 400 varieties that have been released in about 30 countries in Sub-Saharan Africa, South Asia, and some Latin American countries, they are not all expressing the maximum level of the nutrients. So some of these are early releases, and some are not as competitive as the best ones. And so naturally, market forces, farmer preferences will weed them out. And these 400 that are available now might, decrease with time as climate and other stressors take their toll on the performance of these varieties. So 400 varieties, let's say maybe 30% of them have the highest level of nutrient possible and are performing well agronomically. Of course, this adds to the diversity of cultivars, right? It doesn't take away from biodiversity. It just adds to the diversity of varieties available for farmers to adopt as they see it fit for their economy and their productivity. And then climate change, well, 17%, 15% of the iron zinc decrease is being predicted as a result of increasing CO2 levels. And so since biofortified crops have between 50 and 100% more of the nutrient, the impact on the micronutrient content of the biofortified varieties will not be as great. It will still be reduced by that 15%, but it won't be on the basis of 50, let's say, ppm of iron. It will be on the basis of 80 or 90 parts per million iron. So with the biofortification with minerals as a buffer to the potential impact of rising CO2 levels.
You. I think, go ahead. We have another.
Sorry, this might be a naive question. I just wonder why is there no multiple micronutrient for biofortification in single crop?
On which ones?
Why is there no multiple micronutrient for biofortification in single crop? Like fortification of both iron and zinc into one crop.
What about multiple micronutrients in a
Yeah.
single crop?
In a single crop.
Okay, yeah. Well, because the genetic the genetics behind each of the nutrients is very complicated. So for zinc, there are multiple genes involved and breeding for multiple genes at the same time. Just for that one nutrient, zinc can be very time consuming and complicated. So with conventional breeding, that means identifying the high micronutrient materials, then crossing them with varieties that are highly productive to have a highly productive high micronutrient material. It takes a lot of investment and time. So initially it was calculated that developing high iron or high zinc or high vitamin A variety would take between 12 and 15 years. And now that time has passed and now we are just churning out the engine to produce more and more. But it took that long just to, end up with a higher iron bean or higher iron pearl millet, etc. And stacking of the genes that will result in multiple micronutrient advantages would even be more complicated. And that's what the GMO laboratories are trying to do, to stack up several micronutrients. But to date, there are no tangible products from that effort. So the golden rice is supposed to sometime in the future have higher iron and zinc as well. But to date, there is only the golden rice, high provitamin A material, and then the high zinc, high iron rice is in a different pipeline at the same Rice Research Institute in the Philippines. So they haven't been able to put them together and still have a highly productive rice for countries to adopt. So it's really complicated genetically, and that means also economically to breed for multiple nutrients at the same time. The one natural advantage is with zinc and iron that when you increase zinc or iron in a cereal or a legume, you see the other mineral increasing a little bit as well. So that's the case with when you increase zinc in wheat, iron also goes up, and that's the reason for having that efficacy trial in Pakistan. It didn't see an improvement in plasma zinc at the end of the trial, but serum ferritin went up because of the higher iron content of the biofortified wheat, which was not done intentionally for iron, but it just increased enough that there was an impact on the iron. So you had two minerals for one there. Yeah, sorry, I was rambling a little bit, but
Thank you.
Thank you. No, we appreciate that. And I was going to add as a last comment, I think we definitely recognize all the challenges with zinc. We're seeing the same thing with industrial food fortification and all the challenges around how we evaluate what the impacts are. So anyway, we could spend a lot of time discussing that, but I think we're unfortunately out of time. So I just want to thank you again for joining us. Really appreciate it.
thank you for the opportunity. And any questions that come up, that need answering, you can send them to me, and I'll try to answer them with my colleagues.